Okay, so good morning everyone. My name is Zainab Ansari and I'm your biology mentor. So today we are going to discuss structure of nucleic acid from the chapter molecular basis of inheritance, which is of great importance, which has highest weightage in NEET. In NEET 2024 it has given us 6 questions, and in NEET 2023 also it has given us 5 questions. It is very important. Okay, let's start the structure of nucleic acid. Nucleic acid can also be called as polynucleotide. Polynucleotide, as the name suggests itself, it is made up of n number of nucleotides, or we can also call it as nucleotide is the structural unit of nucleic acid. There are basically two types of nucleic acids in living cells. One is DNA, deoxyribonucleic acid, other one is RNA, ribonucleic acid. So we will see everything in detail in both of them. So first of all we will see what is the nucleotide, what it is composed of, what is its composition. So nucleotide is composed of basically three things. First one is pentose sugar. As the name suggests, pentose 5 कार्बन शुगर. कितना कार्बन है पेंटोज शुगर के अंदर? 5 कार्बन है. सेकेंड composition is nitrogenous bases, and third one is phosphoric acid. So nucleotide composed of 3 main constituents: pentose sugar, nitrogenous bases and phosphoric acid. We will see them in detail. We will see the structure of sugar in detail. Pentose sugar, 5 carbon sugar. We will see its structure in detail. Pentose sugar. It is of 2 types actually. Pentose sugar is of 2 types. First one is, we will see first sugar. First one is ribose sugar. First one is ribose sugar, which is found in RNA, ribonucleic acid, ribose sugar. And second one is deoxyribose sugar. So we will see this 5 carbons: carbon number 1, carbon number 2, carbon number 3, carbon number 4, and this one is carbon number 5. Okay, so this is ribose sugar, which I have drawn in front of you. This is ribose sugar. What is deoxyribose sugar, which is found in DNA? Deoxy, नाम से ही पता चल रहा है, ऑक्सीजन को रिमूव किया गया है. कहाँ से? सेकेंड पोजीशन से, सेकेंड कार्बन से. इफ वी रिमूव ऑक्सीजन, then it is deoxyribose sugar. So this is the sugar which is deoxyribose and found in DNA. The sugar is deoxyribose, which is found in DNA. What is the difference between the two sugars? Ribose sugar has OH on its second carbon, but deoxyribose sugar does not have that oxygen on second carbon position. So this is deoxyribose sugar. Now we will switch to second composition, that is nitrogenous bases. This sugar, I hope it is clear. So we will move to second component, that is nitrogenous bases. Nitrogenous bases. Nitrogen containing bases, nitrogen containing compounds. So these are also of 2 types. One is purines, another one is pyrimidines. One is purine, another one is pyrimidines. They are, so nitrogenous bases are of 2 types. First type is purines and second type is pyrimidines. If we see their basic difference, purines are dicyclic. They have 2 rings, and pyrimidines are monocyclic. They have 1 ring. Dicyclic means they have 1 ring and then the second ring. This is dicyclic structure of purines, and this one is monocyclic, only single ring they are composed of. Then what is next difference? Purines have nitrogen on 4 positions, carbon number 1, carbon number 3, carbon number 3, 4, 5, 6, 7 and carbon number 9. So purines have nitrogen on 4 positions, 1, 3, 7 and 9. And in pyrimidines, nitrogen is found only at 2 positions, 1 and 3. Okay. Now there are also different types. Purines and pyrimidines are also different types. We will see their types. Purines are of 2 types, adenine and guanine. And pyrimidines are of 3 types, cytosine, pyrimidines are of 3 types. First one is cytosine, second one is uracil, third one is thymine. So 2 types of nitrogenous bases, purines and pyrimidines. Purines are of 2 types, adenine and guanine. Pyrimidines are of 3 types, cytosine, uracil and thymine. So in totality 4 types of nitrogenous bases we have seen. Now we have to see the combination of these purines with pyrimidines in DNA as well as in RNA. If I talk about DNA, then in DNA 4 nitrogenous bases are found: adenine, guanine, cytosine and thymine. But if I talk about RNA, then it has adenine, guanine, cytosine and uracil. So what is the difference between DNA and RNA? In DNA, thymine is present and in RNA, uracil is present. And if we talk about sugar, in DNA, deoxyribose sugar is present and in RNA, ribose sugar is present. So these are the differences which we should know. First of all, we should know. Now we will switch to third composition of nucleic acid. Third composition of nucleic acid is phosphoric acid. Phosphoric acid, we can write it. This is the structure of phosphoric acid. 3 OH group and 1 double bond group. We can write it also H3PO4. When it combines with sugar and nitrogenous bases, then it gives this H, O negative, O negative, O negative, double bond group. So this is phosphoric acid. So we have studied 3 components of a nucleotide. One is sugar, another one is nitrogenous base, another one is phosphoric acid. Now we have to see how they are bonding together to form a nucleotide. Which type of bonds are there, how they are bonding, at which carbon position of sugar nitrogenous base is attached, at which carbon position of sugar phosphoric acid is attached. So we will see the structure of a nucleotide. This is pentose sugar, this one is pentose sugar, and this is its carbon position 1. On carbon position 1, nitrogenous bases are attached. Here nitrogenous bases are attached. It may be adenine, guanine, cytosine, thymine or uracil. Nitrogenous bases are attached at carbon 1 position with the help of bond named as N-glycosidic bond. N-glycosidic bond. So nitrogenous base is attached on carbon 1 of pentose sugar with the help of N-glycosidic bond. This structure which is in front of you, this structure is called as nucleoside. What it is called as? Nucleoside. It is not nucleotide, nucleoside. Now we will form nucleotide by adding phosphoric acid on carbon number 5 of pentose sugar. This is carbon number 5 of pentose sugar, CH2OH, and we are going to attach phosphoric acid on it. This is phosphoric acid, OH, 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 O. So this is, what is this group? OH, alcohol, and what is this group? Acid. And the bond name which is formed between alcohol and acid is called ester bond. Which bond? Ester bond. So between alcohol and between acid, the bond formed is ester bond. So we'll remove water from here. And after removing water, we will get now the structure will be like this. This is the structure now. So this complete structure now is called as nucleotide. Okay, let's summarize the bond again. Pentose sugar, carbon 1 position is nitrogenous base which is attached with the help of N-glycosidic bond. Pentose sugar, carbon number 5 bonded with phosphoric acid with the help of ester bond. Now because phosphoric acid is attached, so this ester bond will be phosphoester bond. Phosphoester. So this complete structure is a nucleotide, and only sugar and nitrogenous base is nucleoside. So many questions are there on the naming of nucleosides in previous needs. So nucleoside naming is a different topic, how we name nucleoside on the basis of nitrogenous bases. इसके ऊपर बहुत बार क्वेश्चन आया हुआ है नीट के अंदर, तो वो हम डिफरेंट सेगमेंट में देखेंगे. For now we have done with the structure of nucleotide, which is the structural unit of nucleic acid, and there are so many nucleotides in nucleic acid bonded with another bond, phosphodiester bond, that we will see while we will form the bonds between one nucleotide and the another nucleotide. Okay, thank you.